the lipid bilayer, that is the plasma membrane of cells, and it has in the center a hydrophobic nonpolar part. And what happens, because there are these molecules, there are the phospholipids, the glycolipids, and the cholesterol. And they have all something with them which makes them polar at one side and non-polar at the other one. They are the phospholipids. They have a phosphor head. That phosphor group head is polar. And the polar phosphor head group head of the phospholipids is hydrophilic. It is polar and hydrophilic. It loves the water. So it turns to the watery portion on the outside in the extracellular fluid, and a bunch of other ones of them are turning to the inside to the cytosol, the watery fluid part of the cell inside of the cell. And so they're just all there, and we're turning around, all with the head facing the phosphor head, the phospholipids, the watery part on both sides. And then they have just then the tail to the inside, the two fatty acid tail, which are hydrophobic, they don't like the water, they turn away, like magnets turning around. And then in the center there is now this hydrophobic, the tails, the fatty acids, they don't like water, hydrophobic non-polar region. And these phospholipids, they are 75% of all these lipids there. And then the cholesterol, they are 20%. And they have an OH alcohol group at their head. And that forms hydrogen bonds with the polar phosphor heads of the phospholipids. So these cholesterol things are also going with the head, with their OH alcohol group head to the heads of the phospholipids. And they have also a tail with steroid rings and a hydrocarbon thing. And then the last 5%, these are glycolipids. They have a sugar carbohydrate attached, and that is doing the same. It's going also there to that. And that way the lipid bilayer is formed. The glycoproteins, the glycolipids, they're going only to the outside. They're only in the outside membrane, and that is why outside layer. And that is why the lipid bilayer is asymmetric, because, for example, the glycolipids are just at the outside. And that is according to Gerard-Tugan-Sender-Grovski's Principles of Anatomy and Physiology book on the pages 62 and 63. But this is now substitute for medical knowledge and also now medical advice. Hi and welcome. I'm trying to learn cytology. Today, the plasma membrane and the lipid bilayer. And after we've done it, we're going to do extensor carpi or strength training. And if you participate in the extensor carpi or strength training, then it's certainly honest. The plasma membrane. The plasma membrane is a flexible yet sturdy barrier which surrounds and contains the cytoplasm of a cell. Okay, it's around the cytoplasm of a cell. The plasma membrane is around it. It is quite sturdy. The fluid mosaic model describes its structure. Okay, there's some type of fluid mosaic model. It is describing its structure. The molecular arrangement of the plasma membrane resembles an ever-moving sea of lipids that contains a mosaic of many different proteins. So how membrane is shifting, is arranging new. And these are consisting out of lipids, which have many different proteins. The proteins may float freely like icebergs, be moored at specific locations, or be moved through the lipid sea. The lipids act as a barrier to the entry or exit of charged or polar substances, while some of the proteins in the plasma membrane act as gatekeepers that regulate the traffic of polar molecules and ions into and out of a cell. Figure 32, is it helpful? Plasma membrane depicting the fluid mosaic arrangement of lipids and proteins. Membranes are fluid structures because the lipids and many of the proteins are free to rotate and move sideways in their own half of a bilayer. Is it by every membrane so? Because I heard so much about membranes, but I heard never that they're having that were consisting out of fluids, that were a bilayer, and that makes a lot of sense. Lipid bilayer, so we're having at the top with the green balloons over there, and the bottom side, then we're having channels and pores, so through them, okay, that makes sense, there are traffic lines through them, some glucoproteins, some peripheral protein, glucose, polar head, the fatty acids, and the polar head. The fatty acids in the middle, the tails, are hydrophobic and the heads are hydrophilic. Integral protein, cholesterol. So the bilayer, they can rotate, they can switch probably, and they're just like balloons. I think that is not that bad a description. They are just going there like all against the ceiling. And because they're enough, it is no problem if one or two go away, then another one goes away. But it's also possible to go through the balloons. They are not fixed. They can move to the side, but they're still basically closed. This molecular arrangement of the plasma membrane resembles an ever-moving sea of lipids that contains a mosaic of many different proteins. The proteins may float freely like icebergs, be moored at specific locations, or be moved through the lipid sea. The lipids act as a barrier to the entry or exit of charged or polar substances, while some of the proteins in the plasma membrane act as gatekeepers that regulate the traffic of polar molecules and ions into and out of the cell. The plasma membranes of typical body cells are about a 50-50 mix by weight of proteins and lipids that are held together by non-covalent forces such as hydrogen bonds. So there's the same amount of weight of proteins as of lipids. There can be more proteins or lipids, but we're having the same weight. But this is what we're saying there. They are held together by hydrogen bonds. So we're not forming proper new bonds, but the hydrogen is sharing in a covalent bond its electron. And because it is sharing its electron with someone else, it doesn't have a full electron anymore. Just 0.7 or something, 0.9. And because it has just 0.9, it has a little positive charge. And that positive charge is attracting to other negative charges. The lipid bilayer. The basic framework of a plasma membrane is the lipid bilayer. It was actually enough of that. Now, the basic framework of a plasma membrane is the lipid bilayer. So that the thing with the balloons, and one are attracting to the top, other ones to the bottom. The basic is two back-to-back layers made up of three types of lipid molecules. Phospholipids, cholesterol, and glycolipids. Okay, so there. We have the phospholipids. We have the one that we have phospholipids and then it goes down. And cholesterol, we have in the middle, the yellow thing in the middle, little more fancy balloons. And about 75% of the membrane lipids are phospholipids, lipids that contain phosphate groups. Present in small amounts of cholesterol, a steroid with an attached OH, an alcohol group, and various glycolipids, lipids that have sugar carbohydrate groups attached. Okay, there are more things. The bilayer arrangement occurs because the lipids are amphipathic molecules, which means that they have both polar and non-polar parts. That makes a lot of sense. And phospholipids, the polar part is the phosphate-containing head, which is hydrophilic. The non-polar part are the two long fatty acid tails, which are hydrophobic. Because the the phospholipids molecules orient themselves in the bilayer with the polar heads facing outwards. In this way, the polar head faces. It's just on both outside is filled with water. It's just like on the outside is just the water. And the balloon wants to go to the water or so they're going to all directions, and every time the other head doesn't want to go there. I'm not sure if I find an example for the living bird. Doesn't really work out. It's just on both sides is water. And the watery part, the thing with the balloon, goes then towards water, and the tail, which doesn't want to go to the water, wants to go away. In this way, the polar heads face a watery fluid on either side, cytosol on the inside, and extracellular fluid on the outside. On the inside is cytosol. The hydrophobic fatty acid tails in each half of the bilayer point towards the center of the membrane. So the membrane is in the center. It's just the center there because there is the center. There hasn't to be something. There hasn't had to be something. There doesn't have to be something. The hydrophobic fatty acid tails in each half of the bilayer point towards the center of the membrane, forming a non-polar hydrophobic region in the membrane's interior. Is it of help that it is hydrophobic? Is it so that not things can go through? How hydrophobic is it? What can jump through it? Glycolipids account for about 5% of membrane lipids. The carbohydrate groups form a polar head, whereas the fatty acid tails are non-polar. Glycolipids appear only in the membrane layer that face the extracellular fluid, which is one reason the bilayer is asymmetric or different on its two sides. So there are two different types of these phospholipids, there are the phospholipids and the glycolipids. Where are they? Glycolipids. They're looking differently than before. Whatever. One of them is facing only the outside, only the extracellular fluid, not the cytoloid, or what was it? The cytosol on the inside. Why? The remaining 20% of plasma membrane lipid are weakly empathetic. What was it even meaning? So it means that they have both polar. That means that. So like an amphibian, it lives in water and on sea. Like an amphibian vehicle, it can drive on land and it can swim in water. So empathetic. They have one part is polar and another one is non-polar. The remaining 20% of plasma membrane lipids are weakly empathetic cholesterol molecules, which are interspersed among the other lipids in both layers of the membrane. Okay, and then there are the empathetic cholesterol molecules. They're on both sides. Then we have a cholesterol. They are just on both sides. The tiny OH group, so the alcohol group, is the only polar region of cholesterol, and it forms hydrogen bonds with the polar heads of phospholipids and glycolipids. The cholesterol, was there something said to it? The remaining 20% of plasma membrane lipids are weakly empathetic cholesterol molecules. So 1/5 of a plasma membrane are the slightly polar, non-polar cholesterol molecules. They are just on both sides. And the tiny OH group is the only polar region of cholesterol, and it forms hydrogen bonds with the polar heads of phospholipids and glycolipids. That is why it docks on there. That is what we're saying. The cholesterol molecules, they are docking onto the heads of the glycolipids and the phospholipids, which are the balloons at both sides. They're docking onto the head, and therefore they are then on both sides, because they're having a slightly binding to it. Why was it? Because the hydrogen bonds, the slightly forming hydrogen bonds. The stiff steroid rings, where are they? Yeah, here. The stiff steroid rings and hydrocarbon tail of cholesterol are nonpolar. The cholesterol things, the yellow things and that little tail there. The stiff steroid rings and hydrocarbon tail of cholesterol are nonpolar. They fit among the fatty acids tail of the phospholipids and glycolipids. The plasma membrane typically has more cholesterol than the membranes of organelles. The plasma membrane typically has more cholesterol than the membranes of organelles. We had just a little structure thing about organelles. I thought the organelles were inside of the cell. Whatever we mean that, we don't have time. If if you were here for not biochemistry, if you were here for cytology, you can go directly to the next part of cytology. There we will continue with something. If you stay here, participate in extensor cover your strength training in hold and stand standing legs and hands are formed to fists and bring them down. And up fists and again down. And up. And we're having 3 rules. The first rule is that we're stopping immediately as soon as there's pain. The second rule is that we're going only as far as we can go with a little bit of effort. And the third rule is that we're not overdoing it, just to strengthen, not to exhaust. You can use a sock, a t-shirt or towel, press onto it and bring the fist again down. Again up. Change. Up. Down. And change again. Press to the outside. All painless, only as far as it goes. Again in. Press with all 5 fingers equally. Then out. In. Change. Press. In. And again out. And press again. And now down. And up. And down. Up. Change. Press on top. Up. Down. And just press in neutral position. And change. And that was the extensor cover and now strength training part. I thank you very much.